for today is to cover the circulatory system of vertebrates. And as I did on the handout that I gave you last time, at the top I've put our goal. So we're going to cover the anatomy and physiology of the circulatory system with a strong structure-function approach. We're going to do a comparative examination of vertebrate circulatory systems. And then we're going to describe some structure and function relationships. And again, I've been selective as to the topics that we're going to cover in the next hour and 15 minutes or so. There certainly are many more structure-function relationships that I would have liked to have had time to discuss. What was this? So I wanted to start with comparative anatomy of the circulatory systems across vertebrates. All vertebrate organisms have a closed circulatory system. And what it means to have a closed circulatory system is that there is a distinction. Oh, you already have one? Okay, that's fine. There's a distinction between the blood and the interstitial fluid, and that blood is contained within vessels. There is a heart that pumps and generates pressure to circulate blood through those vessels, and then within the vessel system, there has to be a site where material can be exchanged between the blood and the interstitial fluid. Okay, so this is a pretty basic drawing that I made just showing what a basic closed circulatory system might look like. What I want to do is talk about three different circulatory systems across different groups of vertebrates, and we can compare and contrast them and talk about some of the advantages and disadvantages that each one has. This one for you. What we're going to see is that overall, the benefit of a closed circulatory system is the ability to generate more pressure. Okay, and by generating more pressure, this allows the organism to deliver oxygenated blood and nutrient-rich blood to a larger area of tissues and organs that need it. And so this ties in nicely with the second benefit in that a closed circulatory system can meet higher metabolic demands than an open circulatory system can. Okay? Before we talk about the circulatory systems, I have this list of important terms, and what I had initially thought we would do is take really large flashcards and Sharpie markers and write out the terms and write out the definitions, put them up on the board, and play a matching game. Because if you have experienced going through vocab terms with your students like I have, it gets really boring really fast, and everyone winds up being conked out on the desk. So I try to make it a little more interesting sometimes when I can with a matching game, or if you want to take it to the next level, if you're teaching an AP course, you can do some concept mapping. So if you put up some names of specific arteries and veins and the chambers of the heart, you can say, well, which ones carry oxygenated blood? Which ones carry deoxygenated blood? Which of these structures have valves? Which of them don't have valves? And so you can start drawing some relationships that go a little bit beyond the terms. But once you have the note cards made, there's more that you can do with them. So if we had had more time, that was something that I was going to have us do. But I'm pretty sure that most of these terms are basic enough that we can look up the definitions and feel pretty comfortable with them. Is that a fair assumption? Okay. So let's jump into the circulatory system designs then, starting with the circulatory system design for a fish. These particular figures came from the general biology textbook that we use in our university. It's Campbell's Biology. And this is the pretty figure for the two-chambered heart and circulatory system of a fish. On page 2 of your handout, I made my own representations so that we can fill those in. But a fish has a two-chambered heart, one atrium, blood returns to the heart in the atrium, one ventricle, blood is pumped away from the heart by the ventricle. There is one circuit for blood flow. So blood flows from the ventricle into the gill capillaries. Blood picks up oxygen in those gills. The oxygen-rich blood then flows through an artery to the systemic capillaries where oxygen is delivered to the organs and tissues, and then the oxygen-poor blood returns to the atrium through a vein. This type of circulatory system has a disadvantage in that there is only one pump, and there are two capillary beds. So the pressure generated in the ventricle is going to pump the blood into the gill capillaries, but the large increase in surface area in the gill capillaries is going to, God bless you, is going to cause a dramatic decrease in the amount of pressure, God bless you, and so there's very low pressure then to get the blood through the systemic capillaries and back to the heart. Okay. So a disadvantage here is that it's a low pressure system.
What does the second one say right when you need heart?
Ventricle, and then this is atrium. So on your handout, in the left-hand column of here, letter A is gill capillaries, letter B is systemic capillaries, and 1 and 2 are ventricle and atrium of the heart. Okay. Is everyone okay with that? Yes? Good. Okay. Amphibians have a slightly more complex circulatory system. Amphibians have a 3-chambered heart. We have a right atrium, a left atrium, and a ventricle, one single ventricle. So blood is pumped from the right atrium into the ventricle, from the ventricle to the pulmocutaneous circuit, where oxygen is picked up, returns to the heart through the left atrium, passes into the ventricle, and then out into the systemic circulation. Okay? And the advantage here is that there is more pressure generated because the heart is pumping the blood twice. The disadvantage is that there's going to be some mixing of blood in the ventricle because oxygen-poor blood is flowing from the right atrium into the ventricle, and oxygen-rich blood is flowing from the left atrium into the ventricle, and there's mixing of blood taking place. Is everyone good with that? Yes? So in your middle handout, the middle figure on the handout, letter B is the systemic capillaries, C is the heart, and I drew 3 arrows there where you can label right and left atrium and then ventricle. And I'd like to point out that
When you look at the heart,
Yeah.
the left one is the one that you're looking at, or is it?
so you're looking at the heart.
If this was would this be,
Yes, your left.
that's my
Okay.
left.
It's
So it's as if you were the heart.
Right. So when we
We're
look,
looking at your image right now.
when you look at an organ, you're always looking at it in anatomical position, as if it was the patient's left and right, and not your, as you're looking at it. Yes. That's a good question. The other item that I'd like to point out is this figure has atrium labeled A and ventricle labeled V. When we teach our students in lecture and in lab, and when they have to identify chambers, we do not let them abbreviate atrium A and ventricle V, because A could be artery and V could be vein. So we let them use R and L for right and left, but that's where the abbreviations end. We're very specific with them that we don't want them to abbreviate because it then gets tricky. Well, how do I know that you meant atrium and not artery? How do I know you meant ventricle and not vein? So we just say, no abbreviations, you need to write it out. So even though they're using it here, we would not use that with our students. Okay. The last circulatory system here is the mammalian circulatory system. Mammals have a 4-chambered heart, so right atrium, right ventricle, left atrium, left ventricle, a section of tissue separating the right side of the heart from the left side of the heart. There are 2 distinct circuits for blood flow, so blood returns to the heart through the right atrium, passes into the right ventricle, out into the pulmonary circuit to pick up oxygen, back to the heart through the left atrium, left ventricle, and then out into the systemic circulation. Because there is that physical separation between the ventricles, there's no mixing of blood. And so this circulatory system has the benefit of delivering high pressure blood to the organs that need it with maximal oxygen concentration. And this is essential to meet the metabolic demands of mammals. So I've given you some advantages of this circulatory system. Can anyone see a disadvantage or think of a disadvantage? Any thoughts?
It's more complicated.
It's more complicated. It's more complicated. That's one. If you want to build a house, small house versus large house, what are some of the needs if you want to build a large house as opposed to a smaller one? Money. It's expensive. A bigger foundation. You're going to have to have different sizes for your materials. You're going to need more energy. You're going to need more materials. So this is very energetically costly, and it's very material intense. So for an organism like a fish that doesn't have the same metabolic needs as we do, doesn't have the same resources that a mammal does, this would be cost prohibitive, right? But for larger organisms that have more metabolic demands and more resources available, this is necessary and essential. So you have to keep in mind that, well, why wouldn't you want something like this? Well, it's very energetically demanding. It's very resource taxing. Okay? I think sometimes we forget about those aspects when we think about what mammals have, and we take for granted what we have and the energetic demands and the material demands that are required to obtain those things. Okay? So on the far right of your handout, I gave you space to fill in lung capillaries in letter A, the heart in letter C, and then I gave you 4 arrows so that you could label the 4 chambers of the heart. Are we good on that? Okay. I'm going to spend a little more time talking about the mammalian heart. I'm going to trace the flow of blood through the mammalian heart on the PowerPoint slide. Then I'm going to come around to each table and show you a model heart, and we'll trace the flow of blood. And then you have a handout in which we'll fill in the pathway of blood flow through the heart. Okay. As we did with the respiratory system, we're going to use the human as our model for a mammalian heart and for the mammalian circulatory system. So here is an image of a model of a human heart. This is right side, this is left side again. You're looking at a human as if I was standing in front of you. This is my left side. This is the heart's left side. Blood enters the heart through the right atrium, number 1, passes into the right ventricle, passes into the pulmonary circulation. So these vessels here would be arteries. Arteries carry blood away from the heart, and this is always a good time to remind students of some of their misconceptions. I think students always tend to think that arteries are red in our textbook and veins are blue. But it's really, arteries are always carrying blood away from the heart. And in the case of going from the right ventricle to the lungs, that blood is oxygen poor. And blue really means oxygen poor, and red really means oxygen rich. And so that's something really important to get across to your students, is that A is for away. Arteries carry blood away from the heart. So these are pulmonary arteries, and that always trips them up when we do the concept mapping, because they always want to put pulmonary arteries in oxygen-rich blood, but pulmonary arteries carry oxygen-poor blood. Okay, so the pulmonary arteries carry the oxygen-poor blood to the lungs. Then the oxygen-rich blood returns to the heart from the pulmonary lungs through pulmonary veins into the left atrium, into the left ventricle, and then out to the systemic circulation through the aorta. Okay? So I'm going to come around with the model of the heart, and we'll do this again. I'm going to peel the front of the heart off and leave that on the table, mostly because I'm afraid I'll drop it and break it. So, superior vena cava into the right atrium, right ventricle, then it gets pumped. The pulmonary circuit I pulled off, but that would be the pulmonary artery. Out to the lungs. Here's our two pulmonary veins. Left atrium, left ventricle, aorta. Superior vena cava is here, inferior vena cava here. Right atrium, right ventricle, blood would come through pulmonary circuit, which came off. So this is a pulmonary artery. To the lungs, back in through the pulmonary veins, left atrium, left ventricle, aorta. Good? Wow, that's coming down. Glad we're in here right now. Superior and inferior vena cava brings blood into the right atrium. From the right atrium, it passes into the right ventricle, and then it's going to come through. There would be a vessel right in here, the pulmonary artery, carrying the blood to the lungs. These are the pulmonary veins, which return the oxygen-rich blood to the left atrium, left ventricle, and then the blood passes through the aorta. You have a perplexed look.
No.
No?
Just processing.
Okay. Superior and inferior vena cava bring oxygen to the right atrium. Bring blood. Bring blood to the right atrium. The blood then passes into the right ventricle, through the pulmonary artery to the lungs, back through pulmonary veins into the left atrium. Blood gets pumped to the left ventricle and then through the aorta.
So the left has the oxygen-rich blood.
The left has the oxygen-rich blood. The right has the oxygen-poor blood. Yeah. Yep.
This side, then this side is a
Right atrium, right ventricle, out to the lungs.
cool. Okay.
Back from the lungs, left atrium, left ventricle, out to the body.
So it's like when, when someone is, I don't know, like in despair, and people say, oh, you started to turn blue. So would that mean that they're oxygen poor?
Right, so that would mean that the circulation
Right.
to that region is diminished.
Okay.
Yeah. So like when it's cold, my hands always turn bluish
Right.
white.
Right.
And what's happening there is you lose heat. As your blood flows close to the surface of your skin, you lose heat. And so your vessels will constrict when it's cold to try to conserve as much heat. So your blood isn't flowing as much. to those regions, and so
Okay.
they turn blue.
So the atrium is where the blood gathers, it's the ventricle pumps it out.
Yeah, so the atria are the receiving chambers, and the ventricles are the distributing channels. Pushers.
Push out the
Yeah. I mean, the atria still do contract and push the blood into the ventricles, but within the heart itself. Yeah.
Would they put a pacemaker?
They would put a pacemaker in. So your sinoatrial node is your pacemaker, and that's found in the wall of the atrium. So if that isn't pacing properly, it would be replacing the function of your sinoatrial node. Whether they actually put it into there, I don't believe they do. But that's where the natural pacemaker is in the wall of your right atrium.
Chamber, and then,
Well, they're both chambers. These are all chambers. Each one of them is chambers. These are receiving chambers. They receive blood from either your body or your lungs.
okay,
And then the ventricles are pumping it out to the body or the lungs.
and what's pumping it out?
The ventricles.
The ventricles.
Yeah.
Okay.
Okay. So here's the heart. This is your superior vena cava, and this is the inferior vena cava. And they are returning blood to the heart from the body. So this is your right atrium. Superior vena cava. Right, so superior drains the blood from everything that is above the level of the heart. And the inferior vena cava is draining blood from below the level of the heart. Yeah. Draining is kind of a strange word when you're working against gravity. But
Pesky gravity thing, you know.
Now the heart of heartless.
so,
Big Bang.
so blood returns to the right atrium, then passes into the right ventricle. And from the right ventricle, it passes through the pulmonary artery and goes to the lungs, which would be on either side. And then from the lungs through these pulmonary veins and into the left atrium, then into the left ventricle, and from the left ventricle gets pumped through the aorta and out to the body. We good? Yeah?
Yeah, you just need to browse.
Sure. Are you good? This is right atrium, superior and inferior vena cava.
Right atrium receives blue blood.
Mm-hmm.
Okay. I think
And then
we're
this is
good.
the right, and you're good?
I think so.
Okay, good. Okay, so here is the flow chart, and I'd like to give credit where credit is due. My colleague Tara Fay at the university made this handout and shared it with me. We both teach structure and function both semesters, and we share a lot of our materials, we share a lot of our assignments, and we don't team teach, but we do a lot of similar things, so we share our resources with each other so we aren't recreating the wheel, which has been really, really nice to have. So she made this handout and shared it with me. Blood flows from the systemic circulation through 3 vessels to return to the right atrium. Can someone name one of them for me? One of the 3 vessels. From the systemic circulation, so from the body. Superior vena cava. Inferior vena cava. And the third one is returning oxygen poor blood from the heart. Okay. So that would be the coronary sinus. So your heart has its own blood supply. Even though blood is flowing through the chambers of the heart, it doesn't use the blood as it's flowing through those chambers. So there is a coronary circulation. So there is a set of blood vessels dedicated to delivering blood. And so the coronary sinus returns blood from the coronary circulation. Blood returns to the right atrium, passes from the right atrium to the right ventricle. As it's doing that, it has to pass through an open valve. Does anyone know the name of the valve between the right atrium and right ventricle? The tricuspid valve. The tricuspid valve You may also know this as the right atrioventricular valve. Atrioventricular describes where it is, between an atrium and a ventricle. Tricuspid describes that this valve has 3 cusps to it. Okay. We have our students learn right atrioventricular in lab and not tricuspid. Okay? So from the right ventricle, blood is pumped into the pulmonary circulation. And again, it has to pass through a valve on its way to the pulmonary artery. Does anyone know the name of the valve between the right ventricle and the pulmonary artery? Not bicuspid. The mitral valve is the valve between the left atrium and left ventricle. So it's left atrioventricular or bicuspid or mitral. That's that valve. The valve between ventricles and pulmonary circulation is called the pulmonary semilunar valve. And blood will pass then through the pulmonary arteries to the lungs. And I'll give you some time to fill that in. Okay. From the pulmonary circulation, blood returns to the left atrium through a pulmonary vein, passes from left atrium to left ventricle through the bicuspid or mitral or left atrioventricular valve. They're all referring to the same structure. We'll do bicuspid / mitral or left atrioventricular. Valve. Okay. From the left ventricle, blood passes to the aorta, which is the largest artery, passing through an open aortic semilunar valve along the way, and then going to the body. If you had made flashcards for vocab terms, you could have your students take those terms and put them in the right order in a flow chart, or make a map of where blood is flowing. And I tell my students when they're studying, I give them the list of key terms, and if they make flashcards for all of those terms, they can do that on their own. They can use their flashcards more than just for learning the definitions of the terms. Any questions on this? Is this good? Okay. I want to talk a little bit more about those valves that we just identified as we were tracing the path of blood flow through the heart. The job of the heart is to pump blood to the tissues that need it. And so in order to be an efficient pump, blood flow through the heart has to be unidirectional, one way. And we traced that pathway. Heart valves are structures in the heart whose job is to ensure that that blood flow is indeed unidirectional. Okay? We talked on Tuesday about how air flows from where pressure is high to where pressure is low. Well, blood flow is the same. So when your atria are contracting and generating pressure, it's going to force the blood into the ventricles, which is the way we want it to go. But then those atria are going to be empty, the muscle is going to relax, and the pressure is going to drop. Now all the blood is in the ventricles, and the ventricles are going to contract, and pressure is going to build up. And the tendency is going to be for that blood to go from the ventricles where the pressure is high back into the atria where the ventricles is low. We don't want that to happen. That's bad. So what happens is those valves, those atrioventricular valves, snap shut so that blood cannot flow backwards. Okay? Likewise, pressure is generating in those ventricles and it forces blood out into the pulmonary artery and the aorta. That's good. But then the pressure in the ventricles drops when they are relaxed and empty, and the pressure is really high in the aorta because that's where all the blood is. So the tendency is for that blood to flow backwards. That's bad. So those semilunar valves close so that blood doesn't flow backwards. Is everyone okay with that? I'm just going to tell you, I normally spend 20 minutes or so with my students really hammering home that idea. We draw on the board pressure differences. We write down exactly when things open and when things close. If we had more time, we might need to do that, but you seem to get it, so I think we're okay. But we do spend quite a bit of time talking through this. Here are the 4 valves again. Your tricuspid valve is here. Bicuspid valve is here. The pulmonary semilunar valve and the aortic semilunar valve. We talk about the flaps always pointing from atria into ventricles. So they're opening towards where the blood is going, and then they close to prevent backflow. We have these chordae tendineae on the tricuspid and bicuspid valves to prevent the valves from everting, and that helps to keep them closed when they should be closed.
Now, when they replace a heart valve, there for a while they were using pig valves,
Mm-hmm.
then they went to bovine. I guess they only use parts of that. What part do they replace? They take the whole thing out, or do they just the top part where it opens and closes?
So they have to take the valve cusp for sure.
Yeah,
Whether they take the chordae tendineae as well, I'm not 100% sure.
I get,
The papillary muscle here is part of the chamber of the heart, and so I would imagine that would stay intact. But whether or not they replace the chordae tendineae or not, they are attached to the papillary muscle. They're also attached to the valve flap. So something has to be detached and reattached, and I'm not sure exactly where they do that. I wonder if...
I have to teach 7th grade life science. We do anatomy and physiology.
Mhm.
We do people pigs.
Mm-hmm.
But the heart's so small, so I usually
Right.
go somewhere where they'll give me adult pig hearts. And when you open them up, you can really see those
Chordae tendineae.
Yeah.
Mm-hmm.
And they're so stringy, and I can't envision them taking all that,
That out.
obviously they can. But,
Right.
Just, it's amazing to me that, those things really shut.
Right. Yeah, we
valve
di-
itself, but how they would use, like Stan has, he told us he had a leaky valve, and he had, just part of a bovine valve put in.
Okay.
So because I guess the, cow heart's so big.
Okay.
They don't use the whole thing, but I don't know, I wouldn't know what part they misplaced.
I wish I knew. But I know the valve flap
They
for
do it a
sure.
lot, yeah.
Right. Oh,
I would
absolutely.
imagine if it's not closing all the way, but I just wondered if those tendons that pull it shut are the problem.
I'm not sure.
Papillary is where they actually start to branch off, so
Yeah.
my assumption would be
That they would stay behind.
They're up,
from the point of the papillary probably.
Yeah.
That's something to check out.
We can check that out. Yeah. I wonder if it varies from person to person too, depending on how severe the damage is. I don't know. I really don't know.
Yeah, I have a friend that was hit by lightning. Now, they didn't put it, they put, you can hear them clicking.
Oh wow.
They replaced 2. She can't hear them, but when you come close to her,
You can hear them.
click, click, click,
Huh.
click. Yeah,
Wow.
It's really spooky there.
Sounds like my car.
Sounds like my knees. Click click. Okay. One more structure-function relationship in the heart before we move on to blood pressure and blood vessels. I want to talk about the thickness of the walls of the heart. And I'm going to walk around now with this portion of the heart. Remember, this is how the heart sits. I've pulled it off and I've turned it around. So this is right, this is left. This is right, this is left. Right, left. What I want you to do as I walk around is I want you to observe the thickness of the walls of each chamber. The heart wall has 3 layers. The middle layer, the myocardium, is the thickest layer. That is where the cardiac muscle is. What you should observe is that that thickness is not the same in all 4 chambers. And then I'm going to ask some questions about which ones are different. So again, right, left. Right, left. This is right, this is left. This is the right side, this is the left side. Right. Yeah. This is the right side, this is the left side. Okay? Okay, so first question. Which chambers have a thicker myocardium, the atria or the ventricles? The ventricles. Why? Right. The ventricles have more work to pump the blood into circulation, right? To either the pulmonary circulation or the systemic circulation. Okay, so now knowing that the ventricles have more, which of the ventricles has the thickest myocardium, right or left? Left. And why? Has to pump blood through the whole body. Exactly. So that structure-function relationship makes a lot of sense, yes? So if you're ever given just a section of the heart like this and it's sitting on the table, by looking at the wall chamber thicknesses, you should be able to tell that this is the left ventricle and this is the right. And we will do that to our students on a practical. We will cut open a sheep heart and just put half of it on the table and say, which chamber is this? And we have to know, based on wall thickness, that this would be left ventricle. Okay. And this cross-section is just demonstrating the difference in thickness of the wall of the left ventricle versus the right. Okay. We have a little time. So just to talk for a minute about some valve disorders. 2 types of disorders. One would be a stenosis. A stenosis is caused by a narrowing of that opening for the valve. When we met on Tuesday, we talked about flow rates and how flow rates change as the diameter of the structure through which flow is happening changes. So if you have a stenosis, if you have a more narrow valve, there's going to be more resistance as the blood is trying to flow through. And that creates some issues and can be heard through abnormal heart sounds. So normally you would hear lubbed up, lubbed up, lubbed up, lubbed up. Someone who has stenosis of the mitral valve, and again that's the valve between the left atrium and left ventricle, if you were listening you would hear lubbed up whistle, because there's the blood isn't flowing cleanly from the atrium into the ventricle. Insufficiency is when that valve doesn't close properly, and so you get backflow of blood. So it'd be lub swish step, lub swish step, lub swish step from the backflow. Okay, is that? And then I wanted to talk lastly about what happens if there's an interventricular septal defect. So this wall between the right ventricle and the left ventricle is called the interventricular septum. If there's a defect or a hole in that interventricular septum, then there's mixing of oxygen-poor blood and oxygen-rich blood. And I borrowed this figure from Wikimedia. Okay. Okay. We'll do this one next. Okay. As the heart is pumping blood, it is creating pressure. And that pressure is highest in the aorta, and as the distance away from the left ventricle increases, the pressure in the vessels decreases. Okay? The pressure that we measure with a sphygmomanometer and a stethoscope is the hydrostatic pressure, the pressure being exerted by the blood on the vessel walls. And if you take someone's blood pressure, you're going to get two readings, the systolic pressure and the diastolic pressure. The systolic pressure is the maximal pressure in the arteries when the ventricles are contracting. The diastolic pressure is the minimal pressure in the arteries when the ventricles are relaxing. And we always have our students practice taking blood pressure on one another. I've given you some basic steps. So you put the blood pressure cuff around the subject's arm. You want the arm to be relaxed. You can have them rested on a table. You put the stethoscope over the antecubital region of the elbow, so you're measuring the pressure in the brachial artery. You pump up the cuff so that it's at a pressure that's greater than the systolic pressure, and then you gradually let that pressure out. The first sound that you hear corresponds to the systolic pressure, and then the last sound that you hear corresponds to the diastolic pressure. I am a difficult subject to take blood pressure on. Sometimes what's easier is for me to not listen for sounds, but to watch the needle, and you can see like a jump in the needle. And that's the way that most practitioners take my blood pressure, as opposed to listening for the sounds. Yeah. I have lower blood pressure than most. Okay. And again, so here is the pressure in the aorta. We go to arteries, arterioles, capillaries, venules. There's not much pressure left in the vena cava as blood is returning to the heart. Okay? If we had more time, we would talk about some of the factors that influence blood pressure. But just real quickly, cardiac output is the volume of blood ejected from the heart per minute. So that's measured in milliliters per minute. That influences blood pressure. One's blood volume influences blood pressure. So if you have greater blood volume, you're going to have greater blood pressure. And then within a given vessel, the resistance provided by that vessel impacts blood pressure. So if you have a given volume of fluid passing through a smaller diameter vessel, there's going to be more pressure than if you have a given volume of fluid flowing through a larger diameter vessel. So that idea of diameter affecting the amount of space that the item has to occupy. Are we good with that? Okay. This idea that the hydrostatic pressure decreases as the distance away from the ventricle increases, this can be explained in part by the branching that happens when arteries branch into arterioles, delivering blood to organs, and then branches into capillaries to deliver blood to tissues. There's a large increase in surface area, and so that blood gets distributed to many different vessels, and so that increase in surface area causes an overall decrease in pressure because the blood is distributed to more vessels. And even though the surface area decreases from capillaries to venules to veins, there isn't a way to generate more pressure in those capillaries, so you don't see an increase in pressure. Okay? And when we talk about blood pressure and blood flow, we can see that there's a pretty large pressure gradient from artery to capillary, right? So the pressure gradient, we talked about flow being equal to delta P and inversely proportional to resistance on Tuesday. So there's a large pressure gradient that influences flow rate. And then depending upon the radius of the vessel, that will create some resistance to flow. The other two terms come into play here with blood, so the blood's viscosity or thickness, and then the length of the blood vessel. So if we have more viscous blood and more vessels, that will decrease flow. If we have a smaller diameter vessel, that will also decrease flow. I think this is a really nice figure to remind students about the relationship between vessel diameter and flow rates. This came from the general physiology textbook that I use. God bless you. In these two vessels, there was the same pressure gradient, but this vessel has twice the radius of this vessel. So flow rates are 16 times larger in vessel 2 than they are in vessel 1. Why 16 times? Because radius is raised to the 4th power in impacting resistance. If you want to, when you're talking about blood flow and resistance, the activity that we did for airflow with the cups and the water through the straws, you could do here as well. Do you remember that? I've got it here. The 2 cups of different diameters, yeah. Okay. Okay. This should say 4, 5, and 6. But resistance increases as blood passes from arteries to arterioles to capillaries because the diameter of the vessels decreases. And so what does this do to flow rates as blood is flowing from arteries into arterioles into capillaries? It slows it down. Why is that advantageous? Right. So blood flow is slow through the capillaries. Exchange happens at the capillaries. There's sufficient time for exchange to occur because flow is slow. Okay. We're doing well. Okay. Let's talk a little bit more about those exchange vessels, the capillaries. Normally, I talk about structure-function relationships in arteries, arterioles, capillaries, venules, veins. It's an hour or so of material. We talk about all of the structures in each, which has what and not, and the benefits of the structural differences. I've chosen for today to just talk about the structure-function relationships in the capillaries, which are the exchange vessels. Okay? For this, we have to recall back to Fick's law of diffusion. I thought I had fixed that. That diffusion rates are going to be higher when surface area is maximized and when the distance across which diffusion takes place is minimized. The wall of a capillary is well suited for exchange. The wall is thin. It only contains the tunica interna, so one layer of simple squamous epithelial tissue cells. And then there's a basement membrane which provides support. So that's not cellular, it's protein. There are lots of capillaries, so there's lots of surface area for exchange to happen across, and they're thin. Okay. There are 3 different types of capillaries in the body, and I've given you on the handout pictures of all 3 and space to write where in the body you would find these different types of capillaries. The first type are continuous capillaries. And continuous capillaries has, as the name suggests, a continuous layer of endothelial cells lining the tube. There are some spaces where the cells join. They're called intercellular clefts. And so materials can either pass through the plasma membrane of the capillary wall or through these intercellular clefts. We find continuous capillaries in the central nervous system, in the lungs, in the skin, and in muscles. The second type of capillary is a fenestrated capillary. Here we have not only these intercellular clefts, but actually within the epithelial cell itself, there are pores called fenestrations. These fenestrations are between 70 and 100 nanometers in diameter. Nanometer 10 to the minus 9 meter. So these are really small pores, but they allow larger substances to cross more easily as compared to the continuous capillaries. Places where you find fenestrated capillaries in the body, in the kidneys, in the villi of the small intestine, where the absorption of nutrients occurs during digestion, in the endocrine glands, which are secreting larger hormones into the bloodstream, in the choroid plexuses of the ventricles in the brain, this is where the cerebrospinal fluid is produced, in the choroid plexuses and the ventricles of the brain. So again, what you should be seeing here is a pattern. We have larger molecules that need to get into the bloodstream, and so it's necessary to have these pores through which they can pass. And then finally, in the ciliary processes in the eye, it's where the aqueous humor is produced that nourishes the lens and the cornea. The ciliary processes in the eye. have the fenestrated capillaries. Okay. The last type are these sinusoid capillaries. The sinusoid capillaries have very large fenestrations, very wide intercellular clefts, and an incomplete basement membrane. These capillaries have pores and clefts large enough that proteins and sometimes blood cells can actually get through. Okay? So places where you find sinusoid capillaries, the red bone marrow, which is site of synthesis of red blood cells. So those need to get into the bloodstream. Okay. The liver, the spleen, and also some endocrine glands. Okay. So again, throughout the body there are different types of capillaries. The type of capillary that you find is going to be related to what materials need to be exchanged. So there's a structure-function relationship there as well. Does that make sense? Yeah? Good. Okay. I had hoped that we would have time to talk about the different ways in which materials can get exchanged across a capillary. Okay, we're not going to have time to do that except to suffice it to say that there are different ways that different materials get across the capillary wall. And you have the information in your packet so that we can talk about it later if you'd like. Okay? Okay. So we started out talking about the organization of the circulatory system of vertebrates. Then we talked about the heart, which was the pump, the vessels, which are distributing blood to the tissues, and I just want to finish with a brief discussion of the blood itself, so the medium that is transporting nutrients and oxygen to the body tissues. Compositionally, the blood is made up of plasma, about 55%. Red blood cells are the majority of the formed elements, so I said 43.5% by volume of blood. 1% white blood cells and platelets are less than 1%, so to make the pie chart, God bless you, I said, well, 0.5% is less than 1. Okay, so about 0.5%. The majority of the plasma is water, but there are also ions and wastes and hormones and nutrients in that plasma as well. So I found this activity on Pinterest that you can use to visually demonstrate to your students what percentage of the volume of blood is plasma versus red blood cells versus white blood cells versus platelets. I have a 600 milliliter beaker here and sticky corn syrup that we're going to use for the plasma. I think if I had to do this all over again, I would not use corn syrup. Maybe vegetable oil. This is a little too viscous, so I'm going to add some water to it today instead of just pouring straight up corn syrup in here. But I think maybe vegetable oil would have been better. I also pre-marked the points where I wanted to put the corn syrup in so that it would be easier. But you want to fill the beaker or whatever you're going to use about 55% of the way with your plasma. And I'm going to cheat, like I said, with some water. Okay. Okay. And then cinnamon hots for red blood cells. They're the bulk of the formed elements, so you should fill almost to the top with those.
Density created in here.
Mm-hmm. I think I spilled some, so I'll have to clean up later. Not quite as high as I wanted it. Ah. Ooh. It is a waste of good candy. It is. So, then some white blood cells, marshmallows.
That's more than 1%.
And some nonpareils for the platelets. They're small, so 0.5% by volume is more than probably what I put in. It looks yuckier in water than it did in just the corn syrup yesterday because things are starting to dissolve faster. But this is roughly then what you get, and you can sort of show the students. When I used just corn syrup yesterday, there wasn't mixing. It all just sat on the top. There was no dissolving, there was no mixing. So using some water, there's more distribution, so it more accurately shows, I think, that these things are suspended in the plasma. So not only can you show, but it is kind of ugly with the nonpareils melting in the in the water.
Those relative sizes are about right?
You can show relative sizes. It's not perfect, but white blood cells are larger than red blood cells, and platelets are by far the tiniest. So relatively speaking, you can show size as well as volume distribution. But it is sort of a waste of the candy, so maybe you can come up with something else. The only thing I can say yesterday is that when I threw this out, everything was extremely sticky, so I'm not sure you would want to keep it anyways and wash them by hand.
Instead of the
Yeah.
cinnamon heart.
Yeah. Right. The thing that I'm saying is yesterday when I did use the corn syrup, it was so terribly sticky that I had to stand in the hallway and shake to get the stuff to come out, and it was coming out kind of in clumps, so I'm not sure if you would really want to have to clean those poppet beads to reuse them. So if you're, yeah. So I mean, this whole big bag was only $3. So anyways. So you can talk to your students then at that point when you have this to tell them that in the plasma there are ions and there's glucose and there's wastes and there's amino acids and there's other things. So the blood functions as a transport medium, but it also has other important functions too. It's a regulator of body temperature, pH, and water volume, and those white blood cells and antibodies that are in your blood are defending against disease. And then finally, to finish up, I want to talk a little bit about structure-function relationships in the red blood cell. I'm going to give you each a breath saver, not because I think you have bad breath. Please do not eat it yet. You will get to eat it momentarily. They don't sell these individually wrapped anymore. Whoops, I'm sorry. Do you want another one?
No, it's fine.
There you go. There you go. There you go. There you go.
Thank you. Oh,
Don't eat it yet.
okay.
Thank you.
Yeah. There you go. There you go. You get the last one from that pack. Don't eat it yet.
You're that kid. You are that kid, aren't you? Everybody has one.
Uh-oh.
Everybody has.
Yeah, everybody has one of those.
This guy coming in really needs one.
Okay. Don't eat it yet. It's hot in here. I could turn the air conditioning back on. Okay. So everyone has their breath saver. I want you to hold it and I want you to observe the structure. It's a disc. It's narrow in the center than it is on the sides. So it's concave, and it's concave on both of the sides, so it's biconcave. The breath saver is a biconcave disc. This is approximately the shape of a red blood cell. A red blood cell has a biconcave disc shape. Now you may eat your breath saver. Biconcave disc. They used to sell these individually wrapped, and it was very easy for me to find them in the store. They stopped selling them individually wrapped, and I had a difficult time finding them. And so I switched over to cream savers. The benefit of cream savers, or the disadvantage I should say, is that they're only concave on one side, not on both. But you can flip over and say, well, this is what it would look like if the surface area was less, and this is what it looks like. I did those once, but they're not round. They're more oval. No, I did do that one year when I couldn't find cream savers. There was a woman yesterday who said she has her students use Play-Doh to make a round ball and then squeeze it in the middle to make their own biconcave disc. And she said that they don't. She said that they know that if they throw it, they will lose it. So they're really good with it. But the biconcave disc is the shape of a red blood cell. There are no organelles within the red blood cell, so when the blood cells are forming, they lose all of their organelles. Okay. What they do contain within them is hemoglobin. This hemoglobin molecule is what contains the iron, which allows the transport of oxygen. And on the surface of red blood cells are the agglutinogens. So type A blood has the A agglutinogen, type AB blood has AB, type B has the B, and type O doesn't have any. We usually spend 20 minutes or so talking about blood typing and interactions between antibodies and antigens.
Now, there's still DNA, right? Even though there aren't any organelles.
Well, there's no nucleus,
Yeah, I know.
so you wouldn't have DNA in there.
Well, then, when they do,
They can't repair themselves.
yeah.
Red blood cells cannot repair themselves.
Then where did they, when they check people's DNA for crime scenes and stuff, that's the white blood cells?
So they would take, so when you mean when they take blood or when they just take tissue?
When they find blood at a crime scene, and they're looking to compare DNA, are they comparing it and there's no DNA in the red blood cell? Where's the DNA from? White blood cell.
It would have to be from the white blood cells. It wouldn't, it can't be, there's no nucleus in the red blood cell.
Yeah, that's what I thought.
Yeah. So functional benefits of the structure of a red blood cell. It's flexible. And this allows it to squeeze through those capillaries. Because there's no organelles inside, there's lots of volume to carry oxygen. And because of the increased surface area due to the biconcave disc, there's the ability to exchange gases. This is really important. My students will always tell me the increase in surface area allows the red blood cell to carry more oxygen. Surface area is for exchange. The lack of organelles gives volume to carry, right? Am I good with that? Okay. A couple of items related to blood flow and blood flow rates and viscosity. We talked about how if we increase viscosity, we increase resistance, and if we increase resistance, we decrease flow. Someone who is dehydrated, there's less water in here, but the same number of cells. That's going to make the blood more viscous. If the blood is more viscous, it's going to flow more slowly. The heart is going to have to work harder. Okay? And you can show that nicely if you've got something like that. Also, if you have an abnormal increase in red blood cell number, I'm thinking blood doping, right? Taking synthetic erythropoietin to increase the number of red blood cells. You've got the same amount of plasma, but you have significantly more cells, and so that's going to make the blood more viscous. So maybe in the short term there's a competitive advantage, but in the long term you're taxing your heart. You're making it work harder because the blood is more viscous. And so that's dangerous. Okay? Is everyone okay with that? Okay. Lastly, I'm going to tell you what I was planning on doing, but why I'm not going to. I had intended to take this steamer insert and pour to show you what gets filtered and what does not across the capillary. It's really going to be messy because the corn syrup is really sticky. And when I was trying to shake it out yesterday, I think I'll get it everywhere. So if you can imagine me pouring the contents of this beaker into the steamer, the red blood cells, white blood cells, and platelets should stay in the steamer insert and the fluid should go through. Okay, and I think maybe vegetable oil might have been a better option. It's not as thick and it might have worked better, but the corn syrup is just not going to flow. Okay, but you can do something like that.
Something with a little bit bigger pore.
Well, I didn't want the pores to be too big because I don't want the platelets to fall through. So, the platelets are dissolved. Yeah, there's still some of them on the surface, but most of them have yeah, and there's still some in there too. They didn't completely dissolve, see?
Oh, yeah.
They just lost their color, mostly. Okay. Do you have any questions? Good luck with the rest of your day today and tomorrow.